Imagine standing in the middle of, like, a toxic, just completely sun-blasted war zone.
Oh, wow. Okay, setting a scene.
Yeah, right. So picture this. The soil you are standing on offers almost zero water retention, like none. The wind is just constantly sandblasting you with corrosive salt, and the solar radiation is relentlessly trying to mutate your cells.
It's basically the absolute definition of abiotic hostility.
Exactly. I mean, if you dropped a normal agricultural crop, like a tomato plant or an apple tree, into this environment, its cells would rupture. Its whole vascular system would just collapse.
Oh, it would be dead in a matter of hours. Honestly, most complex biological systems simply cannot tolerate those extremes without highly specialized systemic defenses.
Right. But if you've ever walked the beaches of Florida or anywhere in the Caribbean, you've probably just casually brushed past a plant that not only survives this coastal war zone, it actively thrives in it.
Yeah, it really does.
So today we are talking about Coccoloba uvifera, universally known as the seagrape, or uva de playa. And I guarantee you this plant is hiding an incredible array of biological secrets.
It really is a marvel of evolutionary biology. I mean, just a masterclass in adaptation.
So our mission for today's deep dive is to completely transform how you view this ubiquitous beach plant. We are going to elevate the seagrape from, just a simple waxy landscaping hedge blocking your view of the ocean into a wilderness survival superfood, a crucial ecological anchor, and a literal frontier for medical research. Okay, let's unpack this, because to understand a plant that can do all of that, we have to look past the surface.
Right, and what's fascinating here is that the seagrape's true superpower, like the exact biological mechanism that allows it to produce these medicinal compounds and survive that coastal hostility, actually lies not in the plant itself.
Wait, really?
Yeah, it relies on this microscopic underground alliance. It's a partnership that lets it survive where almost no other plants can. But before we get into the subterranean biochemistry, we need to establish exactly what this plant is.
Right, because there is a massive case of mistaken identity happening right now.
A huge one.
The beauty and food industries have completely muddied the waters, and people are getting very confused. So let's properly meet the terrestrial seagrape. Botanically, Coccoloba uvifera belongs to the buckwheat family, the Polygoniaceae family.
Which often catches people off guard.
Yeah.
Because when you hear buckwheat, you probably picture a small herbaceous agricultural crop, right? Maybe a foot tall, used for grain.
Pancakes, yeah.
Exactly, pancakes. You don't picture a towering, salt-tolerant coastal tree.
Exactly. And the seagrape exhibits this incredible trait called phenotypic plasticity. It literally changes its physical form based on the immediate environmental pressure.
Which is such a cool survival mechanism.
It is. Like if you see a seagrape on the primary dune, that high-energy zone right next to the crashing waves, it looks like a sprawling, aggressive shrub. It might only be 1 or 2 meters tall, just hugging the sand.
Right, because the salt spray acts as a natural pruning mechanism.
Yeah, just constantly shearing off the top growth.
The apical meristems get burned back by the wind, forcing the plant to grow outward rather than upward. It's a defensive posture.
But if that exact same genetic specimen is planted in a sheltered inland maritime hammock, just like a few hundred yards away from the wind, it can become a massive multi-stemmed canopy tree. We're talking up to 15 meters high.
That adaptability is the hallmark of a survivor. But regardless of whether it's a shrub or a tree, it retains some highly distinctive morphological markers.
Oh, totally. Let's talk about those leaves.
Yeah.
Because they are unmistakable.
Very.
They are these massive, perfectly rounded, leathery shields, sometimes 20 centimeters across, with these incredibly vibrant red primary veins. And because it's in the buckwheat family, it possesses a very specific anatomical feature called an ochrea.
Yes, the ochrea. That is the diagnostic fingerprint of the Polygoniaceae family.
What exactly does that look like?
It's essentially a papery, sheathing stipule located right at the base of the leaf stem, the petiole, that wraps entirely around the branch.
Okay, so it, hugs the branch.
Exactly. If you are ever trying to identify a plant out in the wild and you see that specialized sheath clasping the stem, you know exactly what family you're dealing with.
That's a great tip. And those leaves are so remarkably thick and waxy, protected by a dense cuticle layer, that early Spanish colonists actually used them as makeshift paper.
Really?
Yeah. They would pluck a leaf and literally scratch messages into the tissue with metal pins. And the leaf is so robust that the spongy cells below the waxy surface hold the indentation perfectly without tearing.
That is incredible. It's a natural notepad.
Totally.
Also important to note its reproductive strategy. The seagrape is dioecious.
Meaning there are completely separate male and female plants.
Okay. Precisely. You don't just have one tree with both reproductive organs. You need a male tree producing pollen and a separate female tree to receive it.
Gotcha.
Only the female tree will produce those famous, hanging, grape-like clusters of fruit. It's a high-energy reproductive strategy for such a harsh environment because it relies heavily on insect pollinators to bridge the gap between distinct plants.
I'm glad you brought up the grapes, because this is where we have to clear up that commercial confusion I mentioned earlier.
Oh, the caviar thing!
Yes. We are talking about the terrestrial tree, Coccoloba uvifera. We are not talking about the marine green macroalgae Coccoloba lentilifera.
Which is completely different.
Completely. But it's heavily marketed right now in high-end skincare and like fancy sushi restaurants as Seagrape caviar or umibudu.
Botanically speaking, confusing a complex terrestrial angiosperm like our sea grape tree with a single-celled benthic macroalgae is, well, it's absurd.
It's completely absurd. I mean, mistaking the terrestrial sea grape tree for the marine algae is like mistaking a pine tree for seaweed just because they're both green and live near the ocean.
Right. It makes no sense.
And it causes real issues. I was looking through a botanical profile on Hawaiian flora, and it noted that tourists frequently get sold these cheap leis made of unripe green terrestrial sea grapes.
Oh, no.
Yeah. The locals call it false mokihana because it mimics the genuine anise-scented Pelea anisata plant. The poor sea grape is just constantly having its identity stolen.
Identity theft aside, the physiological differences between the two sea grapes are profound. That marine algae, the sea grape caviar, survives underwater through a process called benthic osmotic equilibrium.
Okay, break down that equilibrium because that's the key difference.
So the algae lives in an isotonic environment. The seawater surrounding it is incredibly salty. To keep from shriveling up, the algae achieves balance by directly absorbing those exact ions from the ocean straight into its cellular structure.
Oh, so it just lets the salt in.
Exactly. It doesn't fight the salt, it invites it in. That's why when you eat sea grape caviar, it bursts with that salty oceanic flavor. It is an absolute sponge for the ocean's chemistry, packing itself with sodium, zinc, and iodine.
But our terrestrial tree.
Oh, if the terrestrial tree tried that strategy, it would die in hours.
Let's immediately.
Yeah. Coccoloba uvifera actively, violently fights salt absorption. It is engaged in a relentless life-or-death biochemical battle to exclude salt from its vascular system.
Which brings us to the big question. If the plant has to fight that hard just to exist on the sand dune, how exactly is it winning? We established the beach as an area of severe abiotic hostility. Let's dig into the physics of that for a second. The sand offers physiological drought because water drains through it instantly. The sun beats down with intense UV radiation. But the biggest killer is the salinity, right?
Unquestionably. High concentrations of sodium and chloride ions in the sand create two catastrophic problems for terrestrial plants: osmotic stress and specific ion toxicity.
Okay, let me see if I have the osmotic stress part right. Water naturally wants to move from an area of low solute concentration to high solute concentration, right? To balance things out.
Correct. Osmosis.
So if the sand is packed with salt and the plant's roots are relatively fresh, the sand literally exerts a physical pull. It sucks the water out of the plant's roots.
That is exactly right. We measure this in terms of water potential. The salty soil has a highly negative water potential. So a normal plant in that environment doesn't just fail to drink, it actively hemorrhages its internal water into the sand.
Oh, wow.
Yeah, it basically dies of dehydration while surrounded by moisture.
Terrifying. And what about the specific ion toxicity? Let's say the plant manages to get some water inside, but it accidentally pulls in all the dissolved sodium and chloride with it.
Then the second phase of the catastrophe begins. Once those specific sodium and chloride ions penetrate the root endodermis and travel up into the leaves, they enter the cells and begin denaturing vital proteins.
They just break them down.
Exactly. They aggressively compete with potassium, which is necessary for enzyme activation. Within hours, the chloroplasts begin to break down, the plant loses the ability to photosynthesize, metabolism halts, and you get systemic cellular collapse.
So coastal plants that actually survive this gauntlet must have some incredible tricks. How do other halophytes, salt-tolerant plants usually handle this?
Broadly, plant biology has developed a few distinct strategies. You have species classified as obligate recredohalophytes.
Recruitohelophytes, okay.
Think of certain mangrove species. They physically absorb the salt water, transport it up to their leaves, and sweat out the toxic salt through highly specialized excretion glands.
Oh, so you can actually see the salt on them.
Yeah, you can actually see the white salt crystals crusting on their leaves.
That seems incredibly energy intensive. Sweating out solid salt. What's the other method?
You have succulent euholophytes, plants like glasswort. Their strategy is dilution. They allow the salt into their vascular system, but they simultaneously absorb massive amounts of water, storing it in huge swollen vacuoles inside their cells to keep the salt concentration artificially low.
So they just puff up to dilute it. But the sea grape doesn't sweat out salt, and it's definitely not a squishy succulent.
Correct. The sea grape utilizes a much rarer strategy. It is classified as a pseudohalophyte. It relies almost exclusively on root-level exclusion.
Just shuts the door.
Actually, yes. It acts as an impenetrable botanical fortress, refusing to let the salt cross the threshold.
And it does this using the underground fungal alliance you teased earlier. Let's finally get into this.
The secret weapon. The sea grape forms an obligate ectomycorrhizal, or ECM symbiosis, with a very specific, highly adapted fungus called Scleroderma bermudans.
Let's translate that terminology for a second. Ectomycorrhizal. Ecto means outside, myco means fungus, and rhiza means root. So this is a fungus living on the outside of the plant's roots.
Exactly. Unlike some fungi that penetrate deep into the plant's root cells, Scleroderma bermudans wraps around the exterior. And this microscopic shield operates through three highly coordinated, incredibly sophisticated biochemical mechanisms.
Okay, walk me through the first one. How does a fungus physically block salt?
Mechanism 1 is the physical sink. The fungus weaves a dense physical sheath of fungal tissue called a mantle completely around the delicate root tips of the sea grape.
Like a jacket.
Yeah, a really dense jacket. This mantle acts as a biological sponge and a barricade. It actively immobilizes the toxic sodium and chloride ions in the soil, binding them up within the fungal tissue before they can even reach the plant's vascular system.
So it's like a microscopic bouncer standing at the door of the root. The water can come in, but the sodium gets stopped at the velvet rope.
I like that analogy, because while the bouncer is blocking the sodium, it's also actively pulling VIPs inside.
Oh, nice.
That is mechanism 2, the selective pump. The fungus actively scavenges potassium and phosphorus from the sand and pumps it directly into the sea grape's roots.
Why is potassium so critical here?
Because sodium and potassium are chemically very similar, and they compete for the same receptor sites in a plant's cells. If sodium wins, the cell dies. By flooding the plant's vascular system with potassium, the fungus ensures that the vital potassium to sodium ratio remains heavily skewed in the plant's favor. It outcompetes the toxin.
I read a really wild detail about the phosphorus part of this pump. The fungus actually secretes an acid into the sand.
Yes, oxalic acid.
Wow.
Coastal sand is highly alkaline. It is basically crushed up seashells, which is calcium carbonate. In that highly alkaline environment, any available phosphorus instantly binds with the calcium, locking it up in a solid chemical matrix that plants cannot absorb.
So the plant is just starving for phosphorus while sitting right on top of it.
Exactly. So the Scleroderma bermudans fungus weaponizes its chemistry. It secretes oxalic acid into the surrounding sand. The acid acts as a highly localized solvent, literally dissolving that calcium matrix grain by grain.
That's amazing.
It breaks the chemical bond, liberates the phosphorus, and immediately shuttles it into the root network.
It is literally mining the sand for nutrients. Okay, so we have a physical barricade blocking salt and a chemical mining operation pumping in potassium and phosphorus. What's the third mechanism? Because you still haven't solved the water problem. The salty sand is still trying to suck the water out of the tree.
Mechanism 3 is the osmotic adjustment. The fungal shield solves the water potential problem by synthesizing massive amounts of internal organic osmolytes.
Osmolytes, meaning molecules that affect osmosis.
Specifically, compounds like the amino acid proline and various soluble sugars. The fungus aggressively packs its own cellular vacuoles with these osmolytes. By doing so, it dramatically lowers its own internal water potential.
It makes itself saltier.
Exactly. It essentially makes itself more salty, using harmless organic compounds instead of toxic sodium than the surrounding sand.
So it reverses the physical pull. Because the fungus is now incredibly dense with sugars and proline, water naturally flows from the sandy soil into the fungus. And then the fungus just hands that fresh desalinated water over to the Seagrape.
It acts as a living desalination plant. The synergy is breathtaking.
So what does this all mean? If this fungal shield is this highly engineered with Kevlar-like mantles, oxalic acid demining, and osmotic vacuums, is the Seagrape practically invincible? Could you just plant one in a bucket of pure ocean water and watch it grow?
In a word, no.
Really?
It is an evolutionary marvel, but it is bound by biological limits. The exact threshold of that invincibility is something ecologists have studied extensively. Let's look at an experiment published in the journal Terra Latinoamericana. Researchers in Cuba wanted to find the exact breaking point of this fungal shield.
How do you even test that in a controlled way? I mean, out on the beach, there's so many variables.
You isolate the variables. They collected Seagrape seedlings from 3 distinct coastal environments in Cuba: Cayo Coco, Guadalavaca, and Las Coloradas. They raised them in a controlled greenhouse environment to remove the variables of wind and sun, focusing entirely on soil salinity.
Okay, that makes sense.
They subjected the different seedling groups to incrementally hostile levels of salt: 0, 5, 15, and 25 decicemens per meter.
Okay, let's pause and contextualize decicemens per meter, or DSM, because to a layperson, 25 doesn't mean much. How salty is the actual ocean?
Pure seawater sits right around 55 DSM.
Wow. Okay.
So 25 DSM is incredibly salty, about half the strength of the ocean. It's the equivalent of a highly concentrated estuarine salt marsh. For context, most standard agricultural crops will suffer catastrophic yield losses or die if soil salinity exceeds even 4 or 5 DSM.
Okay, so pushing these seedlings to 15 or 25 is essentially torturing them. What did the researchers find? Did the different geographic origins matter?
Origin mattered up to a point. At the moderate stress levels 5 and 15 DSM, the seedlings sourced from Cayo Coco performed significantly better than the others. They retained much more of their original stem length, root mass, and total fresh weight. The fungal shield was holding the line brilliantly.
But what about the maximum stress test?
At 25 DSM, the biological defenses completely broke down.
Just failed.
Across all 3 ecotypes, the physical biomass dropped precipitously. The fungal bouncer was simply overwhelmed by the sheer concentration of sodium. The toxic ions flooded the root systems, the potassium pumps failed, and every single seedling suffered systemic cellular collapse and died.
Wow. So the Seagrape isn't immortal. It is just incredibly adapted to survive right up to a very specific razor-thin ecological limit. And if the ocean surges too high or a drought concentrates the salt in the sand too much, the shield shatters.
It exists on the precipice of survival.
Okay, so if the fungus is acting like a microscopic water purification plant, screening out the toxic sodium, but actively pumping in critical minerals like potassium and phosphorus, where do those minerals end up? They don't just disappear into the wood.
They are heavily partitioned into the plant's reproductive structures, the fruit.
Which fundamentally changes how we view this plant. Because of the sheer volume of minerals being pumped through that fungal alliance, the Seagrape produces fruit with a completely unique, highly concentrated nutritional profile. And this directly impacts both wildlife ecology and human wilderness survival.
We shift from how the plant survives the coast to how the plant can help a mammal survive the coast. Let's examine the nutritional profile of the Seagrape fruit, the drupes.
According to nutritional breakdowns, if you eat a handful of ripe sea grapes, you aren't getting much fat. Only about 0.3 grams of lipids per 100 grams of fruit. You get a respectable energy boost from 18.2 grams of carbohydrates. But the absolute crucial statistic here is the mineral ash content. It clocks in at 1.0 gram per 100 grams.
To a food scientist, an ash content of 1% in a fleshy, high-water fruit is exceptionally high.
Let's explain what ash content actually means in this context, because people might picture like campfire ash.
Right, it's not charcoal.
No. When nutritional scientists test a food, they literally incinerate it in a device called a bomb calorimeter. The heat burns away all the carbon-based organic material, the proteins, the fats, the carbs. What is physically left behind in the crucible is the ash. And that ash is pure, unburnable elemental minerals: magnesium, calcium, potassium.
The exact minerals the Scleroderma bermudans fungus works so hard to pump out of the sand.
Exactly. And this is vital for survival. Imagine you are lost on a Florida beach, or you're just on a grueling, multi-hour coastal hike. The sun is beating down, and you are sweating profusely. When you sweat, you aren't just leaking pure water. You are actively draining your body's internal reserves of sodium, potassium, and magnesium.
The essential electrolytes that regulate your own nervous system and muscle function.
Right. And here is the danger. If you try to rehydrate in that scenario by chugging a massive canteen of pure, filtered fresh water, you can trigger a life-threatening medical emergency called hyponatremia.
Water intoxication.
Yes, because you've sweated out your electrolytes, your blood sodium level is already dangerously low. By flooding your stomach with pure, mineral-free water, you dilute your blood even further.
It's an osmotic crisis.
Exactly. Your body attempts to balance the osmotic pressure by forcing that excess water out of your blood and directly into your cells. The cells physically swell up. In your muscles, this causes agonizing cramps. If it happens in your brain tissue, it can cause seizures, coma, or death.
It is a critical hazard for endurance athletes and stranded foragers alike. Hydration without remineralization is dangerous.
But if you stumble across a fruiting sea grape tree, you have found nature's perfectly engineered sports drink. That 100-gram serving of fruit provides 79.1 grams of pure water, completely free of toxic oceanic salt, but heavily laced with the exact potassium and magnesium ash your body desperately needs to maintain its osmotic equilibrium. You hydrate your cells without diluting your blood plasma.
The evolutionary symmetry is striking. And humans aren't the only mammals exploiting this. Coastal ecosystems rely heavily on the sea grape's mineral load. Frugivorous bats, for example.
Oh, like the lesser dog-faced fruit bat.
Specifically Synopterus brachiotis, yes. Field observations show these bats preferentially target high-ash coastal fruits like the sea grape. Flight metabolism generates enormous heat, leading to massive evaporative water and electrolyte loss.
So they're sweating in the air.
In a sense, yes. The bats specifically seek out the sea grape to maintain their own delicate electrolyte homeostasis while flying in tropical humidity.
The entire ecosystem is literally feeling off the hard work of that underground fungus. Humans, of course, have taken this foraging a step further. We don't just eat the raw fruit. We've developed artisanal processing methods.
The culinary applications are historically well documented. Because the fruit ripens unevenly, transitioning from green to a deep reddish purple, it has a naturally complex flavor profile, balancing sweetness with significant astringency.
People use that to make incredible jams and jellies. But the most surprising use I found was fermentation. In coastal communities, there's a traditional recipe where foragers mash the ripe fruit, skin and all, combine it with cane sugar, and allow wild yeast to ferment it, and it produces an absolute powerhouse of a beverage.
The alcohol by volume, the ABV, can be quite staggering for a wild fruit wine.
Analytical testing puts it at 20.9% ABV.
That's fortified wine territory.
That is incredibly potent. And what blew my mind was the chemical analysis of the finished wine. Even after undergoing that intense fermentation process, the liquid retains a massive concentration of the fruit's original antioxidant capacity. The medicinal compounds survive the yeast.
Which speaks to the sheer density of the bioactive compounds present in the raw fruit.
Honestly, hearing all of this makes me want to grab a bucket, drive down to the nearest Florida public beach, and just start stripping the branches. Between the survival hydration, the jams, and a 20% ABV wild wine, I'm sold.
I strongly advise against that.
Oh, come on.
No, seriously, I have to stop you right there, because acting on that impulse could result in significant legal and financial penalties.
Wait, really? For picking some grapes off a bush?
Yes. We need to discuss Florida statute, section 161.053, specifically subsection 2A. The sea grape is heavily protected under state law when it grows on public beaches and shorelines. It is strictly illegal to harvest, prune, or cut wild sea grapes without a permit from the Department of Environmental Protection.
Why the intense legal protection? Is the plant endangered?
It's not endangered, it's structural. Remember how we discussed the dense root system in the fungal network? On a macro scale, that subterranean architecture is physically gripping the sand dune. When tropical storm surges and Category 4 hurricanes hit the Florida coast, the sea grape acts as the primary biomechanical anchor.
Oh, I see.
Its roots possess immense tensile strength. It literally holds the coastline together, preventing catastrophic coastal erosion from washing public beaches and inland infrastructure into the sea.
Okay, so if I go rip branches off, I'm compromising a hurricane defense system.
Precisely. And furthermore, you're compromising critical wildlife habitat. The broad, leathery canopy of the sea grape serves an essential purpose for endangered nesting sea turtles.
How does a leaf help a turtle?
By manipulating the physics of light. Sea turtle hatchlings are phototactic. They instinctively navigate toward the brightest horizon, which historically is the starlight reflecting off the open ocean.
But nowadays,
today, the brightest horizon is often a coastal highway or a beachfront condo. The dense, sprawling canopy of the wild sea grape acts as a vital light shield. It blocks artificial light pollution from spilling onto the sand, keeping the nesting areas dark so the females can safely lay eggs, and the hatchlings successfully orient toward the water rather than crawling into traffic.
That makes total sense.
Yeah.
So public beach foraging is completely off the table.
If you wish to harvest sea grape, you must do so on private, inland property, and you absolutely must have the explicit consent of the landowner. Harvesting from a cultivated ornamental hedge in someone's backyard is fine, but you leave the wild, dune-stabilizing populations strictly alone.
Good to know. Leave the coastal anchors where they are. But this brings up an incredible pivot in the plant's story. The sea grape isn't just protecting the physical beach from erosion and turtles from light pollution. On a molecular level, this exact same plant contains complex organic compounds capable of protecting the human body from chronic systemic diseases. We are moving out of wilderness survival and into modern clinical pharmacology.
This is where the phytochemical research becomes truly groundbreaking. The very compounds the plant evolved to protect itself from extreme UV radiation happen to be highly active in human biological systems.
Let's start with the visual evidence. When that sea grape fruit ripens, it turns a deep, dark purple. That pigment isn't just a color, it's a chemical signature. It signifies a massive load of anthocyanins. Specifically, researchers have isolated cyanidin-3-glucoside, melvidin-3-glucoside, and pituitidin-3-glucoside within the fruit.
Anthocyanins are water-soluble vacuolar pigments. In the plant, they act as literal sunscreens, absorbing high-energy UV photons to prevent cellular DNA damage. In the human body, they act as potent free radical scavengers.
And it's not just the anthocyanins. The total phenolic content of this wild beach grape is staggering. To put it in perspective, the phenolic density of the sea grape rivals, and in some tests exceeds, the commercially bred Gala apple.
To truly quantify the density of those compounds, researchers subjected the plant to a highly technical procedure called thermogravimetric analysis.
I was reading about this. Thermogravimetric analysis. It sounds like something out of aerospace engineering. What exactly are they doing to the plant?
It's an analytical technique used to determine a material's thermal stability and its fraction of volatile components. In this specific study, researchers took the extracted proteins from the sea grape seeds, placed them in a highly sensitive crucible, and steadily subjected them to extreme, precisely controlled heat, monitoring the exact microgram changes in mass as the temperature rose.
And what happens when you essentially bake these plant proteins?
As the temperature climbed, they observed a massive sudden reduction in the physical mass of the sample, specifically occurring within the window of 174 to 237 degrees Celsius.
Why is that specific temperature window so important? What vanishes at 174 degrees?
That window represents the phenolic cleavage phase. Phenolic compounds are bound together by ester and ether chemical bonds. Those specific bonds violently break apart, they cleave, only when they reach that exact temperature threshold.
Oh, so they just shatter.
Exactly. The fact that the sample's mass dropped so dramatically and exclusively in that temperature zone mathematically proves the incredibly dense structural presence of bioactive phenolic compounds woven into the seed tissue.
Okay, so we have absolute proof that the plant is absolutely loaded with these complex phenolic molecules.
Yeah.
But what do those molecules actually do when introduced into human biology? Here's where it gets really interesting, because the medical applications target two of the most pervasive chronic health issues in the world: inflammation and diabetes.
Let's address the inflammatory response first.
Researchers took crude methanolic extracts of the sea grape berries. They exposed human cellular models to this extract in vitro to see how it affected the inflammatory cascade. And they discovered that the sea grape extract acts as a highly selective cyclooxygenase 2 inhibitor.
Often abbreviated as TOX2.
Yes, the OX2. Let me translate why this specific mechanism is so brilliant, because it fundamentally changes how we treat pain. Think of a standard, over-the-counter, non-steroidal, anti-inflammatory drug, like a standard aspirin or ibuprofen. I like to think of them as carpet bombs.
That's a very apt way to describe it.
Right, because if you have a swollen joint or a headache, the aspirin drops a chemical bomb that wipes out the enzymes causing the inflammation. But because it's a non-selective carpet bomb, it also destroys COX-1.
Which is the constitutive enzyme.
Right. COX-1 is the good guy. It's the enzyme responsible for continuously maintaining the thick, protective mucosal lining of your stomach. So the aspirin cures your headache, but by wiping out COX-1, it leaves your stomach acid free to eat holes in your gastric wall, leading to severe ulcers. It completely lacks targeting.
The collateral damage of standard NSAIDs is a massive issue in chronic pain management.
But the Seagrape extract, it operates like a biological smart bomb. The specific phenolic structure of the extract selectively targets and binds only to the inflammatory COX2 enzyme, shutting down the pain and swelling. Meanwhile, it leaves the protective COX1 enzyme in your stomach completely alone.
The pharmacological data is striking. The selectivity index observed in the Coccoloba Uvifera methanolic extract is statistically comparable to synthetic, highly engineered pharmaceutical drugs like Vioxx.
That's huge.
It really is. It suppresses the synthesis of pro-inflammatory prostaglandins directly at the site of trauma without inducing any gastric mucosal injury. To find that level of enzymatic precision in a wild coastal plant is extraordinary.
A perfectly targeted anti-inflammatory smart bomb forged by surviving a hostile beach. That alone is a medical marvel. But then there's the second frontier, diabetes mellitus.
While the fruit extracts excel at targeting inflammation, the ethnobotanical record shows that indigenous populations historically utilized decoctions of the leaves to treat symptoms of diabetes. And modern cellular assays have recently mapped the exact molecular pathways, proving why this traditional medicine works.
What exactly did they find in the massive, leathery leaves?
The leaf extracts are incredibly rich in specific phenolic acids and flavanol glycosides, predominantly gallic acid and a compound called rutin.
And when researchers introduced gallic acid and rutin into diabetic cellular models, they observed the activation of a highly specific biological cascade known as the PI3K-acted cellular pathway.
This is a major signaling pathway that regulates cell cycle progression and, crucially, cellular metabolism.
Walk me through exactly what happens when the Seagrape compounds flip the switch on that pathway. Because it doesn't just do one thing, it attacks the diabetic condition on 3 separate fronts.
It acts as a systemic metabolic recalibration. First, triggering the PI3K-ACT pathway significantly promotes glycogen synthesis. This means it actively helps the liver properly convert circulating glucose into storable glycogen. It packs the sugar away where it belongs.
That's front number 1, just storing the sugar correctly.
Second, it drastically increases peripheral insulin sensitivity in muscle and fat cells. In type 2 diabetes, cells become deaf to insulin's signal to open up and absorb sugar from the blood. The Seagrape extract essentially repairs the cellular hearing, allowing the body's tissues to successfully pull glucose out of the bloodstream for energy.
And the third front?
Crucially, it suppresses hepatic gluconeogenesis.
Meaning the liver making new sugar.
Yes. In a diabetic state, the liver gets confused and often starts manufacturing new, unneeded glucose, dumping it into an already overloaded bloodstream. The Seagrape compounds send a chemical stop signal to the liver, halting the production of excess sugar.
So it helps the body store sugar properly, it forces the cells to absorb circulating sugar, and it stops the liver from making new sugar. All 3 mechanisms working in perfect synergy. The result is a massive, clinically measurable drop in fasting blood glucose levels.
It is a multi-pronged molecular intervention directly mitigating the chronic complications of metabolic disease.
It is stunning. We have just walked through how the plant's internal chemistry selectively blocks enzymatic inflammation and regulates blood sugar via advanced metabolic pathways. But what fascinates me is that long before humanity understood the PI3K-ACT pathway, or COX2 enzymes, we were already utilizing the Seagrape's protective chemistry in much more immediate, visceral ways. This perfectly bridges ancient ethnobotany with what is currently happening in cutting-edge dermatology.
To trace that history, we have to move away from the fruit and the leaves and look at the structural core of the tree, the bark and the roots.
If you take a machete and score the bark of an older Seagrape tree, it literally bleeds. It secretes this thick, dark red, incredibly astringent sap that historical records refer to as Jamaica Kino. If you take the bark or the roots and boil them extensively, you extract a high concentration of molecules called condensed tannins.
Tannins are fascinating macromolecules. They are high molecular weight polyphenols. In the wild, plants use them as a defense mechanism against herbivores because their extreme astringency makes the plant tissue highly unpalatable.
It just tastes terrible to them.
Exactly. But chemically, their defining characteristic is their ability to form rapid, incredibly stable, insoluble complexes with proteins.
And that chemical reaction binding with proteins is the foundation of the tanning effect. Historically, if a person was in a tropical environment and contracted acute dysentery or severe life-threatening diarrhea, dehydration would kill them in days. It was a massive threat to early explorers and indigenous populations alike.
The ethnobotanical remedy for this crisis relies entirely on the Seagrape's condensed tannins. The precise traditional recipe dictates taking 15 to 20 grams of the raw bark and boiling it vigorously in water for 30 to 45 minutes.
You have to boil it that long to fully break down the cellular walls and draw out the heavy molecular weight tannins into the water.
Correct, creating a potent, highly astringent decoction.
When a patient suffering from dysentery drinks that dark, bitter tea, those condensed tannins travel down into the gastrointestinal tract and immediately make contact with the intestinal mucosa, the lining of the gut. And because tannins aggressively seek out proteins, they instantly bond with the proteins on the surface of the gut lining.
They precipitate the proteins, they cross-link them.
They cross-link the tissue, literally tightening and shrinking the cellular gaps. They form a sudden, impermeable, protective proteinaceous shield over the entire infected gut lining.
It fundamentally and instantly alters the mucosal permeability.
It stops the massive fluid loss immediately, preventing lethal dehydration. But it does more than just act as a plug. Because that new tannin-forged protein layer is so dense, it acts as a physical force field against bacterial pathogens. Nasty bacteria, like toxic strains of E. coli, physically cannot adhere to the intestinal walls to continue the infection. The Seagrape tea halts the dehydration and blocks the infection simultaneously.
If we connect this to the bigger picture, we see a truly profound biological through line. Think about the survival strategies we've discussed. The Seagrape synthesizes a thick, impermeable, waxy cuticle to protect its leaves from losing vital water to the harsh, salty wind. Then, when human beings ingest its bark, the plant's internal chemistry creates an impermeable protein cuticle inside our gut to stop us from losing vital water to infectious dysentery.
It's the same strategy.
The survival strategies, the creation of biological barriers against extreme osmotic loss, are completely transferable across kingdoms of life.
That is incredible. The plant teaches our bodies how to retain water the same way it does. And that exact principle of barrier protection and water retention transitions beautifully from the inside of our guts to the very outside of our bodies. Let's move to the skin. Because topical extracts of the terrestrial seagrape are currently causing major disruptions in modern dermatology, specifically in the fight against photoaging.
This brings us back to the hostile abiotic environment of the beach, the ultraviolet radiation.
Right. When human skin is exposed to intense, prolonged UV radiation, the exact same relentless radiation the seagrape endures every single day on the sand dune, it triggers a massive cascade of oxidative stress within our skin cells.
The UV photons physically damage cellular DNA and generate reactive oxygen species. This localized trauma causes the epidermal cells to panic, overexpressing a flood of inflammatory cytokines, most notably interleukin-1 alpha and tumor necrosis factor alpha.
And alongside the inflammation, the radiation triggers the massive overexpression of a specific hormone, alpha MSH, or melanocyte-stimulating hormone,
which is the cellular mechanism behind hyperpigmentation, the formation of sunspots, and accelerated structural degradation of the dermal matrix, what we collectively call photoaging.
But when dermatologists apply a topically standardized extract of the seagrape directly to human skin, something incredible happens. Those potent phenolic compounds and anthocyanins intercept the cellular panic. They actively inhibit the enzymatic production of IL-1 alpha and TNF-alpha. And crucially, they block the cellular receptors for alpha MSH.
It rapidly calms the localized epidermal irritation, drastically reduces UV-induced cellular senescence, and halts the melanocyte overactivity, maintaining an even, structurally sound skin tone. It essentially lends the human skin the exact same chemical sunshield it uses to protect its own leaves.
But the dermatological benefits don't stop at UV protection. It ties directly back into hydration. Remember earlier when we talked about how eating the seagrape fruit acts as a perfect electrolyte sports drink for a lost hiker, providing potassium and magnesium to balance osmotic pressure?
The physiological concept of electrolyte hydration.
Yes. Well, the modern skincare industry has taken that concept and applied it to the epidermis. They are actively extracting those exact mineral complexes from the terrestrial seagrape to create an osmotic hydration network on the surface of human skin.
The mechanism is elegant. By applying a concentrated matrix of potassium, magnesium, and calcium to the stratum corneum, you actively change the osmotic gradient of the skin surface. The minerals exert a physical pull, drawing ambient moisture into the epidermis and locking it there.
It effectively halts what dermatologists refer to as TEWL, transepidermal water loss,
which is, molecularly speaking, the exact same mechanism the seagrape uses to survive the physiological drought of the sand dune. It is using mineral gradients to hoard water.
Wait, I have to pause here. I need to address the elephant in the room. Because in our very first section today, we spent significant time clarifying the massive taxonomy confusion in the beauty industry. We explicitly stated that marine green algae is heavily marketed as sea grape caviar for high-end skincare, and that people shouldn't confuse it with our terrestrial tree. But now you're telling me that this terrestrial tree also works perfectly for electrolyte skincare? No wonder the consumer market is so totally confused.
The consumer confusion is entirely validated. It is one of the most highly unusual coincidences I have ever seen in the commercial botanical space. You have two completely unrelated organisms. One is a single-celled benthic marine algae. The other is a massive terrestrial angiosperm tree. Both are colloquially called seagrape. And astonishingly, both happen to utilize incredibly potent, highly effective osmoregulatory minerals for cellular hydration.
They both do the exact same thing for human skin.
They do. They just source their chemistry differently. The marine algae absorbs its ions passively directly from the isotonic seawater. The terrestrial tree meticulously, aggressively filters its minerals from the toxic sand through its specialized fungal network. But when either organism's extract is applied topically to the human epidermis, both provide profound, verifiable osmotic hydration.
That is just wild. The beauty industry stumbled blindly onto two completely different biological organisms, mistakenly called them by the exact same name, and through sheer impossible luck, they both actually work for the exact same dermatological application.
It goes to show that nature often has more than one evolutionary pathway to solve the exact same biological problem.
Well, looking at the time, we have covered an immense amount of ground today. We took the Seagrape, Coccoloba Uvifera, and we completely flipped the script. We watched it transform from a basic beachhead into a master of survival, shielded by subterranean fungal armor. We saw it act as a vital ecological anchor, holding our coastal dunes together against hurricanes. We explored its role as a potent source of wilderness survival hydration with its heavily mineralized electrolyte-packed drupes, and we unlocked its status as a pharmaceutical treasure chest, armed with complex tannins and phenolic smart bombs, capable of treating deadly dysentery, regulating diabetes, and selectively shutting down inflammation.
It serves as a powerful testament to a core biological truth. Extreme, hostile environmental pressures force the evolution of extraordinary biochemical solutions. The plant is a chemical genius because the environment demanded it.
Exactly. So next time you are walking on a public beach in Florida or exploring a Caribbean shoreline, and you feel those unassuming, massive, waxy, red-veined leaves brush against your legs, take a second to truly appreciate what you are looking at. You aren't just looking at a shrub. You are looking at one of nature's most sophisticated, heavily fortified biochemical laboratories, quietly fighting an unending war against the salt and the sun.
And exploring that laboratory leaves us with a compelling final thought. We've spent a lot of time today mapping the complex fungal network, the Scleroderma bermudans, that acts as the invisible bouncer, mining phosphorus and keeping the sea grape alive in toxic sand. But it makes you wonder, what other invisible microscopic alliances are thriving right under our feet in the most hostile, overlooked, seemingly barren environments on Earth, just quietly waiting to yield the next great medical breakthrough?
A brilliant thought to end on. Keep looking closer and keep questioning the world around you. Thanks for joining us for this deep dive.
Until next time.